guess everyone's here. So, oh, you will start. Okay, great.
Okay, good afternoon, everybody. Welcome to the PhD defense. Albert Kejun Ying is our candidate for the defense today. First, I'd like to thank the committee members who will evaluate Albert's defense. Brendan Manning, thank you very much, Brendan, and Jessica Laskisu here as well, and also Curtis Hattenhauer on Zoom, and also Daniel Belsky from Columbia, also on Zoom. So thank you, everybody. So let me say a few words about our candidate, Albert. He received his bachelor's degree in China from Sun Yat-sen University, but he also had experience, research experience at UC Berkeley and University of Washington before coming to Harvard for a PhD program. So he actually joined at our lab in 2020 during COVID, and this was very interesting because his beginning of his rotation coincided with the beginning of the pandemic, and so he had actually virtual rotation, and he started as an experimental person, but during this rotation converted to computational. He never went back, remained computational. And what is most remarkable to me is that during his rotation already, he proposed an idea of causal biomarkers. It was completely his idea that actually made the base of his thesis. Actually, we decided to expand on it or work on it and expand on it, and it is a major part of the thesis. But although now become much larger because there have been some other causal studies that they're completed and published and then expanded in many other directions, as you will see in the presentation. So in parallel, he also obtained a master's in computer science from Harvard while being a PhD student. Applied for F99 fellowship, which is he's funded now. Would work, but anyway, so he received a perfect score of that very competitive fellowship, and he's also quite amazing in organizing like collaborations. So somehow he's able to bring together people from various labs and in universities and even countries seamlessly. Like they all work together under his leadership or in collaboration, and this results in many studies, collaborative studies. I went today, actually, I myself being a PI, I don't know how many, just too many. I went to Google Scholar, I counted over 30, and then on PubMed 20, but 10 of them is Albert is the first author. So it's quite remarkable for a graduate student. Actually, I've never seen anybody having like published that many papers during PhD thesis. It's really kind of fun stating. So, and the thesis itself is the longest I've seen. These 9 chapters, too many papers. I don't know the committee. I guess had hard time to read. It's too long. But to me, it's very impressive. So with this, I would like to bring Albert to the podium and deliver his presentation today.
Thank you, Vadim, for the introduction. And so I guess I should just show this slide when Vadim introducing. So it's basically, so yeah, basically Vadim just introduced. I was an experimental person at first, and then I converted to computational after I joined Vadim's lab. And so I've been in many labs before I joining, and but all of my experience, they kind of have a shared theme, which is just aging. I'm always very curious about aging, and or it's very passionate about it. So, but why study aging? So I'm from a program in School of Public Health, so I will explain in a public health perspective. So there's a book, very famous book called "Cancer: The Emperor of All Malady." So if the cancer is the emperor, the aging is the fundamental cause of almost all the diseases. So if you look at all the chronic disease, they all associate with aging. And from pandemic, we also know even the infectious disease can also be associated with aging. So this is quote from the business world. So if you cannot measure it, you cannot improve it. So the same should go to aging. So if you cannot really measure aging, it will be very hard for people to manage aging. So in the field of the aging field, there's a concept we call it biological age. So basically, we believe that different people, due to various reasons, they can have different rate of aging. Some people may age faster, some people may age slower. So you can already see from sometimes you can see from face. So sometimes you meet someone and say, "Oh, you look so young." So that's what happened. So we believe there's a latent variable called biological age, which is hidden behind your chronological age, which is your age shown on your passport. And some people, due to maybe genetic reason, or maybe they just have healthier lifestyle, so they sometimes they have lower biological age than their chronological age. So they usually behave younger, look younger, and also have less age-related disease. And some people, unfortunately, also similarly due to maybe bad lifestyle or genetics, they can also have accelerated biological age. So the question is, how do we really predict or measure this thing? So luckily, in 2013, a scientist called Steve Horvath, he found that the DNA methylation mark are highly correlated with the chronological age. So basically, you can just measure this small chemical modification on your DNA and just measure their abundancy. You can find that there's some like DNA methylation site, their level are highly correlated with age. So from this graph on the right side, each line is a methylation site, and the y-axis is their level. So you can see some of sites are consistently have increased methylation level during aging, and some sites have like consistent decrease. So from this information, some people can build very accurate machine learning algorithm to just predict the age of individual, either human or mouse or other animals. So here's one example in mouse. So the y-axis is their age predicted by DNA methylation. The x-axis is the age, the real age in months. So you can see in this blue line that they like the methylation predicted age is highly correlated, actually almost the same as their chronological age. And this age is even responsive to the interventions. So for example, calorie restriction, which is a very famous anti-aging intervention, animal with this intervention are shown to have like younger methylation age, which probably means that this methylation age is capturing something from the biological age. So here I will give you an overview of my thesis and also how I structure my defense today. So I divided it into 3 sessions. The first session would be the causality and genetics of aging, where I will explain more in a minute. And another second session is advanced aging clock, which includes chapter 5 and 6. And last session is a standardization and community, which is in chapter 7 and 8. Okay, so so the first session will be based on following work. So this is all the 3 work I started at the very beginning of my PhD. So I was before I joined my PhD program, my only like computational experience before was on quantitative genetics. So of course, my first computational project will be around this topic. And so all 3 projects are kind of related to genetics and somehow related to causality. So the first question we're asking is that how since we all know like causal relationship is very important when we study biology, can we really establish causal relationship between aging and diseases? And as Vadim introduced, I joined lab while COVID happened. So my first project is actually a COVID project. So luckily there's so traditionally, the gold standard to establish causal relationship is a method called randomized clinical trial. So basically, you have a group of people, you randomize them into 2 groups. So this is one group for intervention and one group for control. And this step is really like crucial for the identifying causal relationship because this step can kind of randomly allocate all the confounding variables across two arms. So all the comparison below that will be independent to any confounding variable if the randomization works. So which means that the difference between the outcome of the intervention arm and the control arm will be the estimated causal effect size. But the problem is also very straightforward. So it's very hard to have a randomized control trial, and sometimes it depends on intervention. Sometimes you have ethical problem. And luckily, in genetic world, we have a method called Mendelian randomization. So this is use similar principle. So basically, you still have a population. But the idea is that your genetic variants are randomly allocated during the inception. So this step actually mimic the randomization step of the clinical trial. So imagine for example, one of your parents carry one mutation on your gene, and you will have like one half of the chance to receive that. And this step is totally random. And then all the people are naturally be randomized into two groups: the people with genotype A or genotype B. And then we just compare between these two arms. Our estimate will be a proxy to the causal effect. So in the first project, we investigated how aging or how lifespan can have causal effect on the risk of COVID-19 and the risk of being hospitalized once you got COVID-19. And this is the result. So basically, each single dot here is a genetic variant. And so basically, for example, in this plot here, if the genetic variant on the right side means that people carrying this variant have longer lifespan, and if it's below, it's on the so y-axis is representing their effect on COVID-19. So if they're lower, means that those people have lower risk of COVID-19, and similar to this one, which is related to hospitalization. So you can see all the genetic variants that associated with longer lifespan, they tend to have effect on decreasing the COVID-19 risk. And a genetic variant that shortening lifespan associated with a higher COVID-19 risk. So based on calculation, the risk of COVID-19 is decreased by around 69%, and the risk of hospitalization is decreased by 54% per 10-year longer parental lifespan. And this is the estimated causal effect. So this is the main take-home message of this first part of the session one. And the second, then since we know that DNA methylation can be used to track aging, one very straightforward question after is that does the DNA methylation change actually causally related to aging? So we use very similar approach, but make it epigenome-wide. So we did this Mendelian randomization approach for all the CpG sites across the genome on the 450K chip. So in total, 450,000 DNA methylation sites. And we did the same way. So basically, there are some genetic variants that naturally occur will lower the methylation level of certain CpG site. So we use them as an instrument to conduct this causal analysis to see whether there are certain DNA methylation sites can have potential causal effect on your lifespan-related phenotype. So this is what the result look like. So basically, we found like roughly a few thousand of CpG sites that has estimated causal effect on lifespan-related trait. So for example, we have, so the y-axis here is the estimated effect, and it's just like a Manhattan plot. So our top hit is actually one methylation site here on Huntington, which previous study shows that this is kind of related to the head bone mineral density. So it's kind of maybe related to the fertility or like bone mineral density, etc. And we take one step further. So so there was always debating like, you have all this change during aging. Are they all like bad, or are some of them adaptive? How do we even separate them? So from the result, from the previous result, we actually obtain the estimated causal effect of each CpG site on a lifespan based on the Mendelian randomization. So we have so if we put it on the x-axis. And we also know how each methylation site change with age. So we can put it on y-axis. So we can really separate all the methylation site into this 4 quadrant and also one area for like no effect. So if you have methylation site, that higher level will promote longer life. And you also observe an increased methylation level during aging. We'll classify it as a protective or adaptive. Similarly, if you have a methylation site that prolongevity and decrease during aging, we call it this kind of change is deleterious. So by this way, we can basically separate all the methylation site into 2 groups. One group is kind of protectively change during aging. Another group is deleteriously change with aging. So based on this information, we can inject this information into the epigenetic clock model, and using this way, we built 2 very unique clock. One we call damage, which representing the age-related damaging related change, and one we call adaptage, which representing protective change. So all these 3 clock, also one cause age, which is just all the site we identify as causal. So all these 3 clock, they look like super similar, just as if you look at their predictive power. So the y-axis here is their predicted age, x-axis here is their real age of a human. So you can see all the clock, they perform like quite similarly. But when we look at the association with mortality, you can see that it's quite striking. So the y-axis here is their effect, a significant score for their effect on for the mortality. So you can see the damage have strongest association with the mortality, and it's actually promote mortality. Means that people with higher damage, they tend to have a higher risk of dying. And conversely, although damage looks similar when you just look at their prediction on age, they actually have inverse relationship in terms of predicting mortality. So people with more adaptive change during aging, they actually tend to live longer. So this is also actually, I think it's the first time we build a biomarker like track the aging, but have inverse association with mortality. Okay, so the last part of this session will be the mutation. So all 2 projects before I mentioned are rooted on the common mutations. So we're using the single nucleotide polymorphism that commonly occur in the population as an instrument. But how about the rare damaging mutation? So in this work, we're trying to investigate what's the effect of the rare damaging germline mutation affect longevity, especially extreme longevity. And previous study have identified like many common genetic variant associated with longevity, and people also find some rare variant that promote longevity. But the role of the burden of the damaging mutation, which we identify as a loss of function mutation, are largely unexplored. So here we use a very large centenarian cohort with whole exome sequencing data. So we have in total 637 centenarian, 900-ish offspring, and 600 support controls. And basically, we quantified their rare loss of function mutation in their genome, and we perform a burden test to see whether people whether like centenarian really obtain lower in total damaging mutation compared to the control group. So basically, this is the result. So so basically, we classify the damaging mutation in several different ways, which is if we only count of the loss of function mutation, which is usually causing a truncation of a protein, or predicted damaging missense mutation using several algorithms. But across almost all the category, you can see the centenarian tend to have lower mutation burden in their germline compared to the control group, and also even their offspring, they tend to have lower, but the effect size is smaller, but still significantly lower mutation burden. And we also perform the gene-based tests to identify there's basically several genes that occurs in centenarian that tend to significantly have less loss of function mutation. Okay, so the summary. So basically, for this session, we established a causal relationship between actually parental lifespan and COVID-19. And we show that people with every 10-year increased predicted lifespan, they actually will decrease the COVID-19 risk by almost 70%. And we identify several potential causal CPG sites across the genome using the Mendelian randomization approach. We create several causality-based clock, and we also found that fewer loss of function mutation centenarians. And so all the paper in this session is actually already published. So a causal clock is actually featured on the cover of Nature Aging last year. And you probably can read more. There's actually one of my committee, Dembelski, wrote this view piece for the causal clock project. So it's actually very super nice. I cannot even explain myself like this clear. And also early this year, last year I also wrote a small like introduction script for Nature Review Genetics about this work. Okay, so this is the end of the first session. And the second part would be advanced aging clocks. So as Lord Kelvin said, to measure is to know. So this is based on these two work I mostly done like last year. And actually the first one that just started like 3 years ago, but I just got finished last year. So the question before I already introduced the concept of biological age. So now the question is, like all the biological age or estimate we have right now, all the aging clock we have right now, they provide a single number. Like basically, if you are 46, I predict your age to 38. What does that mean? So that aging is so complex, does that even can be described by a single number? So we're trying to approach this, and my answer will be no. I think biological age is just too complex as a concept to be represented by a single number. So previously, actually mostly this year and last year, people have exploring this idea at organ level. So this is work last year from Tony West-Corey lab, where they show that they can build aging clock for different organs separately. And they found that some people can have age acceleration in certain organ. And also some people, unfortunately, all the organ, but still it shows that you can have different age at different organ. And this below graph is from work from our lab, led by Luger. So basically it shows that people with older, like certain organ also tend to have a certain like organ-related disease. For example, if I have an older liver, you tend to more likely to get this liver fibrosis. And if you have older lung, you tend to have more risk for COPD, etc. So from this perspective, aging is really heterogeneous across your body. And now here they explore the organ level, but what about biological function? So I propose that just like your organ, different organ can have different age, maybe a different biological function, different functional module can also age differently. So here we try to dissect the biological age concept in the molecular pathway level. So basically the idea is very simple. So across the genome, you have several genes that correspond to certain function, which form into this functional module. Sometimes people call it pathway. And actually most of the time people call it pathway, sorry. And we can just segregate your genome to for a different pathway, and we just take the DNA methylation site representing or inside each of the individual pathway. And we take this methylation site separately, and then we train aging clock just separately for each pathway. And then for both human and mouse, of course. And then we can get a very large collection of the aging clock. In total, I trained 1,863 clocks. And And then we also did many downstream analysis. So we compare their predictive power. We look at evolutionary pattern. We look at the effect on intervention and mortality, disease prediction, etc. So one very interesting thing we first show is that if we look at the methylation rate across mammals, so here the y-axis here is the methylation rate. So previous paper they found that the genome-wide methylation rate actually scale with the mammalian lifespan. So you can see that short-lived animal like mouse, they have quite fast. Of course, this is scaled methylation rate. So we just set the rat to be 1, and they have fastest methylation rate. But longer-lived animal, they tend to have lower methylation rate. So basically, it's very easy to understand. So short-lived animal, the methylation pattern change faster, and long-lived animal, their methylation pattern change slower. And by segregating the genome into different pathways, we also find there's a heterogeneity across different pathways. So here, each pathway, each line is an individual pathway, and we color them by the significance score, which is minus log 10 p-value based on the perturbation. You can see some pathway, they significantly kind of more responsive to lifespan. So people with long-lived, animal with longer life, they tend to have much slower methylation rate on this kind of pathway. But there are also some pathways that kind of ill-evolved. So all species, they tend to have like almost equal methylation rate on those pathways. And more interesting is that those pathways are conventionally thought aging-related pathway. So we have hypoxia, which is kind of very related to aging. We have this ESR-mediated signaling, which is kind of related to estrogen, I think. And like myogenesis, apoptosis, etc. Estrogen, yeah, related gene expression. And we also look at the performance of the this almost 2000 clock on the disease prediction. So basically, we can see that so those dots with black surrounding are the we call reference clock, which is well accepted public clock before. So we can see we can find several like pathway clock they significantly outperform the existing clock. For example, this is for cardiovascular disease. We have several clock that outperform the traditional clock. And more strikingly, it's for cancer. So for some reason, all the published clock, they kind of suffer from predicting cancer. Basically, people with accelerated aging, they don't, it's basically just not predictive to cancer. But here, when we segregate them into pathway level, we can find several kind of meaningful pathway that can really strongly predict cancer. For example, we have one like it's related to cytosolic DNA sensing, which is kind of related to cancer. Also, we have one pathway related to mitotic G2 phase, which is clearly cancer related. So those pathways shows much stronger power in predicting cancer risk. So conclusion here is that by segregating the aging pattern of different function module, we can give us a more complete view of aging and also more information. So we can move from a single number to a distribution of age. And this paper is currently on bioRxiv. Okay, so let's jump to another theme. So, so we all be very like impressed by the recent progress on the large language model. So the recent year is really you see many large language model be trained, and their ability is really amazing. And one of their powers actually come from understanding the context. So based on, for example, if I give you a sentence like something, you are such an amazing person, this could be a real compliment, or it could be a sarcasm, just depends on what I said before. And similarly for DNA methylation, you see some pattern in your DNA methylation site, maybe in your clock, which is just which you only use a subset of the DNA methylation site. but their effect might also be context dependent. So maybe if there's something wrong in the other part, maybe like, for example, one obvious example is that if you just make something so wrong that just kill the animal, well, just selectively make certain species site in your algorithm that shows super young, you will get a young signal, but the animal actually old or dying. So we believe that for DNA methylation, the context might also matter. So the question is that, can we build a model that can understand the context, just like a large language model that can understand context in a sentence? So the large language model uses a mechanism called the attention mechanism. So basically, when they visit each word in your sentence, they know which other word they should refer to. And they will have this attention metric, which given different weight for all previous word, and they will refer to other word to understand the context to explain the current word. So we're trying to do a similar thing for DNA methylation. And we built what we call foundation model for DNA methylation, where basically we collected 150,000 DNA methylation sample from human, varying from for like more than 20 tissue type, and very diverse sample collected from Geo, EVOS Data Hub, Clockbase, which is a database we curated before. And because we have limit on contact length, we pre-selected roughly 50,000 physiologically relevant DNA methylation sites. By physiological relevant, we mean that those CpG sites are previously identified to associated with several outcomes based on the EVOS data. And this gives us in total 7.6 billion training token. And then we train a GPT style model, which is basically we take the methylation data, we make it into a methylation embedding, and then we train it with a mask language model approach. So basically for each part of input, we randomly mask. So all of these squares here are DNA methylation site. So here we randomly mask like 30% of a methylation site. And then we let it go through this transformer layer, which is we're trying to learn what other methylation site they should refer to predict the masked methylation site. And then the model predict the masked site, and we compute loss function based on the difference between the prediction and the ground truth. And by training this way, model will know that when they refer to certain CpG site, what other important CpG site they should also look at. And then we perform some fine-tuning on domain-specific data and so on and so on. But the idea is that once you train a large model, you can use it to almost anything else. You can fine-tune it for almost any downstream task. So the first thing I want to show is that just by this training approach, we can make the model accurately predict the methylation value. So if we just focus on this graph, so the x-axis here is the ground truth DNA methylation. The y-axis is predicted DNA methylation. Each dot here is an individual DNA methylation site, and of course it's a test data. So basically, you can see even though it's masked, the model can very accurately predict the DNA methylation value of the max methylation site. And so if I didn't mention before, the DNA methylation value is actually ranging from 0 to 1. 0 means totally unmethylated. 1 means completely methylated. So we have a median error of 0.07, which is quite accurate. And another thing this kind of large model can offer is the embedding. So by embedding, I mean that basically during training, we represent each individual CpG site as a vector. So basically a series of number. And during the training, the model will learn like what this series of number should look like to represent the relationship between different CpG sites. And so what we did here is that we just extract this contextualized CpG embedding for this almost 50,000 DNA methylation site. And we map them to, we perform the dimension reduction and look at how they look like in this 2D space. So each dot here is representing one single CPG site, and they are clustered based on their embedding number. So we're trying to understand how this embedding, what this embedding related to. So we look at several features. So first is what's their relationship with the CPG island. So you can see here, although it's kind of a little blurry, but you can see kind of clustering for certain kind of island-related CPG. For example, you have shore here. You mostly have other and shelf, which are CPG sites that far away from the island. And you also you have island right here in the middle. And we also look at other features like enhancer region. You can see enhancer are mostly located on the right side of this graph. And if you look at the chromosome, you can see somehow the sex chromosome is kind of clustered together at the middle. So basically, we show that by pre-training, we can get a very meaningful embedding information from the pre-training. And beyond CPG site embedding, we can also extract embedding for our samples. So by similar way. And so this is basically, I just did similar thing for each sample. We have embedding, and we can look at their distribution on this 2D space. So if I didn't do this pre-training, if I just use the raw DNA methylation data, the data would look like this because we collected our data from so many different sources. So you have like very strong batch effect. So usually experiments from same batch will cluster together. And here we color by tissue and see it's really not clusters. But after pre-training, somehow the model kind of internally adjusts for this batch effect. So although not perfectly, but still much, much better than the raw embedding. So you can see like we have kind of mainly brain tissue cluster here. We have mainly blood tissue cluster here, etc. And we then fine tune it to do the age prediction. So and then we compare with several like established method and other published clock. And we can see that we can show that our model can on the same training set and same splitting strategy, we can have a better performance compared to other methods. And this is not striking, but what is more striking is that when we gradually taking out the information from the data. So as I mentioned before, the model internally understands the relationship between the CPG site. So when we randomly mask, mimicking the data missing. So what we did is we just randomly mask the input DNA methylation value to missing data. And we start from 0, which is full data, and then we mask it to like 10%, 20%, 30%, and to up to 90%, which is 90% of the data are representing as missing data. And you can see our model, their performance is kind of very responsive to this masking. So if you have fewer DNA methylation sites, your model will perform like significantly worse. So the x-axis is the error, the y-axis is the error. But for our model, since it's kind of understand internally, it can kind of do the imputation internally. So even though we mask the DNA methylation up to 70%, we can still get meaningful prediction. So this is really useful when you have a data with a lot of missing data. And what I showed before is it's just a tiny model. We trained before with only 3 million parameter, and we now have 2 other larger model with 7 million parameter and 15 million parameter. And we can show that we can see some kind of scaling law here. So model with larger model tend to have. This is also the age prediction task. So we can see larger model tend to have a lower error, and our strongest model can even have a prediction like error below 2 years. So it's very super accurate. So for summary, we built this pathway level aging clock to capture functional aging dynamic. We built this aging representation, and we show that they can improve disease prediction. And we built this one of the first foundation model for DNA methylation site, for DNA methylation of DNA methylome. And we show that we can by reading the context, we can generate very meaningful embedding and very efficient for downstream tasks. So for extended reading, I would recommend this opinion piece right by Eric Topol earlier this year. Basically, he reviewed all the recent he called large language model for life, and our model is one of them. And also there are many amazing models here. And our model is actually currently open source right now. And now we end the second session, so time is good. And the last session would be the standardization and contributing to the community. So for this part, I mainly just working with the this biomarker of aging consortium, which Madi and Jesse initially founded. So this work will based on like 3 other preprint: the clock-based, which is a large database we built, a toolkit we built for predicting methylation, predicting a biological age, and a competition we hold to encourage people to train better model for predicting age. So basically, talking about community contribution, basically they are just a lot of things you can do, but the most important thing, especially for computational people, would be data. So the first question is, can we collect all the data and can we standardize all of them? So here, this is a work I started probably 3 years ago. We call it clock-based. So basically, the idea is that academia have accumulated such large amount of the omic data. So each individual lab, they measure the omics, they upload their data to public database like Geo, and the data just sit there unless some people are interested in this specific data set, and they will download it and do the analysis, et cetera, et cetera. But most of the data just sit there. So the idea is very straightforward. Since we have this omic-based model to predict the biological age, can we just download all the data and just predict the age for all of them and build a large database people can easily access? So here we start from this idea. We build this, what we call clock-based, where basically we take more than 50,000 data sets from Geo, in total up to 2 million samples, including both human and mice, both DNA methylation and gene expression data. And we kind of standardize them and pre-calculate their age prediction based on various aging model. Actually, it's more than 30 different aging clock model. And then we put it into a large database. So, so this is what the data look like. So this is just for the human data. So again, I compressed it into a 2D space just for visualization. So each dot here is an individual sample. And here is colored by predicted Horvath's age, which is actually one of the first epigenetic clock published. So you can see that we have like, you can see many patterns here. For example, the placenta seems to be super young. All the placenta seem to be here. And you have some like infant tissue here, also super young. And you also have some old tissue like differentiating hypothesite, for example, et cetera, et cetera. And based on this, another thing to standardize is the metadata. So if you ever used Geo before, you probably experience this frustration. So you download the data and you realize they have like weird structure of metadata. And if you, especially when you want to use multiple data sets, you will realize different people, different lab, they have a different way to organize their metadata. And it will take a lot of time to do like manually cleaning up. So what we did here is we take advantage of this large language model. So we just give the unstructured text, which is just the abstract of the Geo entry and also the unstructured metadata. And we give them to GPT-4 to ask them based on a template to make a standardized large metadata table. And the idea is that, so basically this is already done. So just we haven't put it on the website yet. So we did this for all of our 2.5 million samples, and now they're basically it's in a clean format. You can just access this large table for all the information like sex, age, disease condition, et cetera, et cetera. And this is a showcase of what this data really look like. So this is also, again, still like human data, and each dot here is an individual sample. So this is a subset of all the data where we have age information since we standardized them before. So the color here representing their difference between their real age and predicted age based on damage I developed before. So you can see some people, they show like extremely old pattern compared to their young age, and some people, they show extremely young pattern from their own age. The idea is that how can we learn any meaningful biological thing from this large data? So this is what we are currently working on here. And so, as I mentioned before, mostly this community-related effort, I work with this large consortium initiated by Madi and Jesse, called Biomarker of Aging Consortium. So it's a really amazing organization. And basically the goal is to making a consensus for the biomarker aging by defining what is biomarker of aging, how do we validate them, how should we develop new biomarker, et cetera, et cetera. So another thing I built for the community is a toolkit for the methylation age, for the biological age in general. So so right now there are so many aging biomarkers scattering around. So what we did here is that we built a centralized platform called BioLearn. It's a Python library. So basically you can very easily query the aging clock and have very standardized interface to just compute the aging clock based on whatever data you have. And we currently curated over 30 biomarker and 20 data sets. And we even allow us to did a quite large benchmarking analysis on this on the data. And the last thing is the challenge. So last year we started this we call Biomarker of Aging Challenge. And we have totally $200,000 award. So for the phase 3, we encourage participants to build a model to predict just chronological age. So we have a data set we generated in-house. We didn't release them before the end of the challenge. So we just let people train model, and we test it on our internal data set to see whose model is best. So our phase 1 is chronological age prediction. Phase 2 is mortality prediction. Both phases are already ended. And our third phase is to be started. It's on a health span or age-related disease prediction. So for summary, we standardize a large amount of sample and large amount biomarker to be inside database. We provide a unified toolkit for computing the age. Just mention also we use the BioLearn, which is a toolkit as a back end of the clock base, our database. And we host this first open competition for biomarker of aging. And for next level of clock base, we want to use AI agent to automatically making meaningful biological discovery from the standardized data. And so you can read more. So in 2023, NBT News is actually featured our clock base. And last year in Science News is featured our competition. And so it's great. So we are toward to the end. So before I reach to a conclusion, I want to discuss one very interesting topic I always thinking when I'm working on all this project. So all of my projects are kind of surrounding on one single concept. It's called biological age. But what is the biological age? It's kind of not really well defined. It's like saying something like what's your luck or what's your like intelligence, right? It's kind of undefinable. Of course, for intelligence, you can kind of arguably can use IQ, which is a test to identify, but still it's controversial. But what about biological age? How do you really define biological age? And this remind me with this quote. So what is time? If no one asks me, I know what it is. If I wish to explain to him who asked, I don't know. So I think a similar thing happened to biological age. So let's rethink this problem from first principle. So just a reminder, like all the biological systems. I know most of the people here are biologists, so some of them you may get offended, but but in deep principle, every biological system is just a very complex physical system. So they still obey the physical rules. So basically, there's no fundamental difference between a organism and a chair, a table, a machine, etc. So this means that then we can ask different questions. So is biological age representing a universal concept to all physical systems? If so, can we find an equivalent to what we call biological age for a non-biological system or even simpler system? And if we can do that, then we can do this backward and to understand what this really biological age is. So we go back to the most simple system, which is a random walk model. So it's super simple. You have a dot in a line, and each time point is randomly jumped either to left or right, and totally random. And it's just keep jumping. And in this case, the state of the system, which can be described by a single parameter, which is x, which is current location of this dot. And my question is this system age? It's clearly like changing with time, right? So if you plot out their trajectory, you can see at beginning all the dots are located at the original point, and with time come by, they kind of going around, and you can see they become wider and wider, and clearly this system changes with time, and clearly they're aging. So okay, so let's just call it aging for now. So we call it aging. So then let's find the equivalent of the concept of biological age in this system. So before that, what's the chronological age of this system? It's very simple. It's just the x-axis here. So the x-axis is recording the time. So the time spent on the system is the chronological age. But what is biological age? So here, the biological age, we can define a biological age equivalent by the expected value of time t given by the state x of the system. So for each of point, since all the information we have is their current location, we just work backward to say, okay, this is my current location. What time did I pass? So this prediction or this expectation, the time t given by x, is your biological age equivalent. So for this kind of simple system, you can even compute this analytically. It's kind of just x squared. Okay, so good news is that the state of biological system is also definable, also not measurable. So actually it's a kind of cheating, but for any physical system, you can just simply define their state as among all the particles, what's their current position, what's their current momentum, and what's their current potential. And you do this for all the particles. The set of this is the state of this complex system, and same for the biological system. So you can simply define a biological state as this, and then although not measurable again, but then you can kind of formalize it. So you can write the concept of biological aging in a very simple equation like the t given by your biological state, and that's it. So you can really define it and just cannot measure it. So why it's useful? It kind of can help us answer so many questions that people are asking. Like there are 10 different aging clocks. In my case, 1,800 aging clock. They all give me different number. How do I interpret them? Oh, 2 clock give me 2 number. The clock doesn't work. So in the fundamental aspect, they are just each aging clock they just measuring different things in a simple word. So you have your biological state, which is unmeasurable, but it can have projection. So it can project to many measurable traits like your ohmic, like your grip strength, like your walking speed, like your lifespan, etc. So different clocks just use different kind of projection and different measurable trait and just calculating this fundamental biological age from different angle. And basically each clock fundamentally just use a subset of this complete system state. And each different clock just fundamentally just capturing like this expected time given by this partial biological state given by the observation you choose. So this could help us really answer the question like this. Okay, really conclusion. So yeah, it's a long defense, but first we went through all the mutation and genetic approach to identify causal relationship with COVID example for disease. We look at the genetic of a centenarian, look at the causal effect of DNA methylation. And we also built two advanced aging clock. One is a platform, a framework called Agium, which the first time we have a system with more than 1,000 clock. And we also have, first time we have this transformer-based model for aging clock. And we built several data set, a toolkit, a challenge for the community, and will hopefully help people to study aging better. And lastly, we're trying to define the biological age from first principle, and kind of we can define it, although we cannot measure it again, but still I feel more comfortable when I work on something like definable. And with this, I want to thank people. So I have 3-page acknowledgement. So, but first I want to thank the people in Gladyshift lab, especially Vadim Gladyshift and other people in the lab, several alumni. And I also want to thank my dissertation advisory committee, Brendan, David, Shamil, my defense committee, Brendan again, Jessica, Dan, and Curtis, my program, my grant, other funding, the core collaborator for session one, which is mostly near Basai, Steve Horvath, and people from Shashin and Peter Federship lab. And our session 2 is mostly people from Bowan's lab, Ricardo and Daniel from Generation Scotland, et cetera, et cetera. And for AI agent, and also the whole biomarker of aging consortium. Some mentee have fortunate to mentored before, and all my previous mentor from my undergrad and lab I rotated. My next step mentor during postdoc will be David Baker and Tony West-Corey. And my whole PhD cohort, they are very supportive during all this 5.5 year. And all the friends and peers, and also, of course, also my families. So thank you so much.
Presentation, but maybe we'll have a few questions from the audience.